Madrid and Director of International Association for Cannabis Medicine, Manuel Guzman.
Ok, boa tarde. Muito obrigado a Laura, a Fernando, todo o comitê organizador pelo convite e por organizar este simpósio. É prazer muito grande para mim estar aqui com vocês. Agora vou trocar o portunhol, vou passar a inglês. Well, in our lab at Complutense University of Madrid, most likely you know, we started to work in cannabinoids by 25 years ago. We started to work in cancer, and Cristina Sanchez, who is around, you know her very well, was the first one conducting that research. She was a PhD student at that time. And then we started to add other lines of research to our lab. We did some stuff on metabolic control, cannabinoids, metabolism, cannabinoids, and brain development. And in the last years, we have also started a new line of research that is focused on neuroprotection. You know that this is a topic that is very much in debate nowadays. There is a lot of research, a lot of mostly preclinical, unfortunately not so much clinical research on cannabinoids and neurodegenerative diseases. And this is also a very debated issue in the media, in documentaries, in internet. Whenever one enters into a website of cannabis, it's very likely that one finds cannabis is neuroprotective, especially CBD. When one goes to one of those so many CBD shops in internet, it's always the claim that CBD is neuroprotective. Well, we're going to see what we know about this. And of course, before entering into the talk, you know what we are dealing with, neurodegeneration. Degeneration is one of the main challenges of modern biomedical research and clinical research. You know that neurodegenerative diseases are very frequent in our environment: Alzheimer's disease, Parkinson's disease, multiple sclerosis, amyotrophic lateral sclerosis, ataxias, etc. Those diseases are devastating, are really malignant diseases, and really strong diseases, debilitating diseases. And nowadays, you also know, we have no cure for those diseases. Do we have even some therapies that can temper the symptoms of the diseases? But we don't have any therapy so far that can cure a neurodegenerative disease, or not even that can inhibit the progression of the disease. So the challenge is very, very strong, and especially because aging is the main risk factor, you know, for these diseases. And the world population is becoming more and more age, is becoming older, and in the next 25 years, it is estimated that the incidence of neurodegenerative diseases will be double, at least double compared to nowadays. So if we find any compound, any therapeutic intervention that can not cure, but at least chronify or inhibit the progression of neurodegenerative diseases, we will really have a superstar in the field of biomedicine. So the task is extremely challenging, but what do we know about this nowadays, and what we expect that cannabinoids can offer us in the future? So I'm not going to repeat what other speakers exposed this morning about cannabinoid and endocannabinoid system, but just a few key questions that I would like to bear in mind. Cannabinoid receptors and overall the endocannabinoid system is very widely spread all over the body, especially in the brain, where it controls a large number of different processes. And perhaps one of them is the control of neural survival, the survival of our nervous cells, of our neurons. Usually, the cannabinoid system is considered to be a silent system. It is not very active in normal conditions, but when there is a disease, this system tries to recover the balance, the homeostasis of the organism, to make it coming back to normality. So it's, by principle, a system that is activated by stimuli, that is activated by trauma. Silent in normal conditions, it's active in disease, trying to get the organism back to normality. So this is what was also shown by Janos before. and Christina, this is a system that, evolutionarily speaking, is expected to protect our body by allowing it relax, eat, sleep, forget, and overall protect from trauma. So I'm going to present you what we know about cannabinoids and neuroprotection in preclinical models. And that is something that I want to state very clearly. We are not mice. We are much more complex animals than mice. So we researchers do studies in mice, in rats, and then we try to extrapolate to humans. But it is not so simple. We are not just big mice. We are much more complex beings, and therefore our pathology, our diseases are much more complex than the diseases that we model in mice. So I will show you 5 lines of research that support that in preclinical studies, in preclinical models, cannabinoids can have neuroprotective actions. We have contributed to this as well as other laboratories all over the world. First, let's go to the molecules. Let's go to the very basics. You know that many cells have cannabinoid receptors, and in the brain it's mostly cannabinoid receptor type 1 on their surface. This is the membrane of a cell, in this case neural cells, that is neurons. And here is the extracellular medium where cannabinoids are. They come from the outside, of course, or they are endocannabinoids also produced by nearby neurons. And here's the intracellular part. Here is the cytoplasm and all the organelles of the cells that recognize this information that is transduced by cannabinoid receptors into different molecular signals. Of course, I don't want you to learn this slide by heart. In this slide, I just want to show you, as a scheme, that we know nowadays what is the mechanism by which cannabinoids, for instance, THC, when it binds to CB1 receptor, can inform the cell, the inside of the cell, that the cannabinoid is there. And some of these responses, these chemical responses, I will tell you about a couple of them now, can, in fact, we know in this preclinical system, favor the growth and the survival of neurons. So from a molecular perspective, yes, cannabinoids elicit different signals inside the neurons that help neurons to grow and to survive. Just an example. When THC or another cannabinoid binds to CB1 receptors, we know that in the inside of the cell, there is a protein that has 3 different subunits. One is called alpha, one is called beta, and one is called gamma. And this heterotrimeric, because it has 3 different subunits, protein can trigger the activation of different proteins. For instance, this protein that is called ERK, or that protein that is called AKT, are some of the most important pro-survival proteins that our neurons have. So yes, cannabinoids have specific mechanisms to enhance the activity of these pro-survival molecules inside the cell. Not only that, cannabinoids do not only act by themselves via cannabinoid receptors. There are other receptors on the surface of neurons that improve, that enhance, that facilitate neuronal growth and survival. For instance, this family of receptors that we abbreviate RTK. These receptors can bind growth factors, other factors, not only cannabinoids, other factors that are present in the medium that induce neuronal generation and survival. So we know nowadays that cannabinoid receptors do not only trigger the activation of these pro-survival signals, but also they have some mechanisms to facilitate the action of these other pro-survival receptors. So in sum, cannabinoids acting both by themselves, that is via cannabinoid receptors, or by facilitating the action of other receptors that also trigger pro-survival signals, can favor that our neurons grow and are more healthy. So we have a relatively good knowledge from the mechanistic point of view about cannabinoid receptors on the surface of neuron being able to favor the growth and the survival of these cells. So point 1 is okay. Cannabinoids can trigger these signals. Let's go a little bit upper. we were talking about molecules. Now we go to cells. In the brain, it's not only neurons that are standing there. There are other types of cells. So what do we know about whether cannabinoids act only in neurons, also maybe on other cells, and whether those processes are also favoring neural survival? That is, they are inducing neuroprotection. The answer is yes. I will tell you in advance, but let's see a little bit how this works. There are essentially three processes in the brain that are favored by cannabinoids that induce neuronal survival at the end. Let's start with neurons. Cannabinoids can trigger, by the molecules I showed you before, what we call an antioxidative action. They can reduce oxidative stress. You know that oxygen is essential for life, but also it's an enemy of all of us, because whenever we breathe, whenever we induce our cells to do respiration and to oxidize different substrates, we are also generating some molecules that are very, very aggressive to our tissue, to our brain tissue. They are called reactive oxygen species. So cannabinoids, by the processes I showed you in the previous slide, can decrease the generation of these reactive oxygen species. They can prevent the oxidative damage of neurons. So this is one process that can lead to neuroprotection. So first, cannabinoids are antioxidant compounds. Second, cannabinoids are what we call anti-excitotoxic compound. What is this? Cannabinoid receptors, both present in neurons that are releasing neurotransmitters, and also in neurons that are receiving those neurotransmitters, and especially one neurotransmitter that can be deleterious for neurons in large amounts, that is called glutamate. Cannabinoids, we know, have the capacity to decrease glutamate release and glutamate action, especially when glutamate is very high. So cannabinoids, by inhibiting the overaction of glutamate, can reduce this process of toxicity in neurons as induced by overexcitation, what we call excitotoxicity. So cannabinoids do not only prevent oxidative damage in neurons, but can also prevent excitotoxic damage of neurons as induced by over-glutamate action. So, good point for cannabinoids. Another good point for cannabinoids. And a third good point for cannabinoids is that on other types of cells that are present in the brain that are called microglial cells, cannabinoids can reduce the toxicity that these cells can exert over neurons. Microglial cells, most likely you know, are the immune cells of the brain parenchyma, of the brain tissue. They are resident macrophages that stand within the brain, and in principle, they have the mission to protect neurons. But when microglial cells become overactivated, they become just the opposite. They become not foes, not friends, but enemies of neurons. And that is what we call neuroinflammation, when a microglial cell is overactivated. So on microglial cells, we have type 2 cannabinoid receptors, not CB1 receptors, as in neurons. We have CB2 receptors, and when cannabinoids bind to these CB2 receptors, we know that they can change the profile of microglial cells to make them good, that is, to produce anti-inflammatory mediators, and decrease the aggressive profile of microglia to decrease, therefore, the generation of pro-inflammatory mediators. So, together, cannabinoids can decrease the toxic overactivation on microglial cells and indirectly induce neuroprotection. So there are, in summary, three different processes, three different cellular processes that we know they occur in mice: decrease oxidative stress, decrease excitotoxicity, and decrease neuroinflammation, that are therefore positive for the brain and can induce neuroprotection in mouse models. In fact, when we use mouse models of neural disease, we usually observe that cannabinoids are neuroprotective. So when we give cannabinoids to a mouse that is suffering a certain neural disease, or when we remove cannabinoid receptors from the mouse brain, and therefore we make the mouse more susceptible to neurodegenerative diseases. And this has been observed in a very large list of neurodegenerative disease models. Both acute brain damage, for instance, in hypoxia, when the brain doesn't have oxygen, when there is a mechanic trauma on the brain of the animal, just heat on the brain of the animal, the skull of the animal, when there is excitotoxicity, as I told you before, when also oxidative stress, and we saw before, those types of damage that we usually call brain damage, they occur in few seconds, but therefore, but despite that, the damage that is prolonged for many hours, days, months, or even years in humans, and they are very, very aggressive stimuli to the brain, different aggressive insults to the brain. But also in chronic neurodegeneration, in long-term neurodegeneration, for instance, in Alzheimer's disease, MS, PD, Huntington's disease, ALS, etc. In mice, we see that cannabinoids are really good to prevent neurodegeneration. So all these aspects, all these different types of diseases really make us suspect that cannabinoids may have a really important neuroprotective role, not only in mice, but perhaps in other species, and ideally in humans. Unfortunately, translating what we know in mice to humans, as I told you before, is extremely complex. It's a great challenge, and especially in the brain. That is a very sensitive organ and a very complex organ and very difficult to study. So all these preclinical studies that are very promising for human treatment, for human therapy, unfortunately haven't been so far translated into mice. In mice, we know that there are no, as I told you before, no therapy that is able to induce neuroprotection in any of these diseases. We don't know any good therapy, and we have to admit that, not only with cannabinoids, but with any drug that we have tried before, or with any therapeutic intervention that we have tried before, to induce neuroprotection in these different diseases. The field is really orphan of good treatments. In a recent review about Alzheimer's disease, that is maybe the best known neurodegenerative disease, in mice, it was listed up to 300 different therapies that were effective to treat Alzheimer's disease in mice. 300 different therapies. How many do they work in humans? 0 so far. So this is what I want to convey to you. It's not so simple to see that cannabinoids work in mice. They work very well, but there are other many therapies that have worked in mice that do not work in humans. However, cannabinoids are not so bad. They are doing relatively well in neurodegenerative diseases. They cannot cure a disease. They cannot prevent the cause of the disease, but they can effectively relieve the symptoms of the disease. And this is not so bad. I mean, all the therapies we have nowadays for neurodegenerative diseases, they are only palliative therapies. We have nothing, as I told you before, that inhibits the cause of the disease, and of course, we don't have anything that cures a neurodegenerative disease. And you know, of course, that MS-associated symptoms can be fairly well treated with cannabinoid, spasticity, nocturia, tremor, neuropathic pain, etc., and also pediatric epilepsy, that Philip and you all know, he showed before, and we know very well, can be effectively treated with CBD. But still, we don't know whether cannabinoids neuroprotect. We know that they palliate symptoms. We have another piece of information in favor of us, and that is when we analyze the brain of patients that have suffered different diseases, different neurodegenerative diseases, we see that there are indeed alterations in the expression of endocannabinoid system elements in different human populations that suffer from brain damage or neurodegeneration, not only in humans, but also in animal models of disease, but most importantly, of course, in humans. Usually when a person suffers from a neurodegenerative disease, and we can have access to postmortem brain tissues, and we analyze the endocannabinoid system in those patients that have died because of neurodegeneration, we usually see that in those pieces of brain, there are more endocannabinoids that we usually interpret as a mechanism of protection, as an adaptation for protection against the damage. We have usually a decrease in CB1 receptors in neurons that we believe make neurons more susceptible to the damage. And we usually observe an upregulation of CB2 receptors in those microglial cells that we usually interpret as a mechanism of adaptation for blunting, for attenuating microglial-induced neuroinflammatory damage. So yes, our brain changes when we have a brain problem, when we have a neurodegeneration or a brain damage. But in spite of that, our endocannabinoid system intends to protect us, intends to defend us, but at the end, it is not so strong to make it, at least only from the endogenous action. And maybe we have to attempt to help from outside. But so far, as I told you, no clinical trial has been conducted in this respect. So as a scheme to take home, as a take-home message, we know that in preclinical models, endocannabinoids via CB1 receptors in neurons, especially neurons, and CB2 receptors in microglia can exert neuroprotection. This is the picture we have today after many years of research in this field. How do cannabinoids or endocannabinoids via cannabinoid receptors produce neuroprotection or protection of our brain or of our central nervous system? First, as I showed you, we know that neurons contain CB1 receptors and cannabinoids can induce protection via different mechanisms that we know well from the molecular perspective. We know also that microglial cells, by blunting their activation, can contribute to this protection of neurons via CB2 receptors. We also know that there are other types of cells in the brain, I didn't show it before, but just for simplicity, such as astrocytes and oligodendrocytes that also contain CB1 receptors and can contribute to this protection of the brain. And we also know that in the cells that generate neurons upon proliferation and differentiation, what we call cellular precursors or cellular progenitors, neuronal progenitors in this case, we can induce the generation, the genesis of neurons and the genesis of glial cells from activation of CB1 and CB2 receptors. So altogether, via combining CB1 and CB2 receptor-mediated actions on these cells, especially on neurons, but also on bystander, on accompanied cells in the brain, we can induce the protection of the neurons. However, as I told you, the evidence we have so far is preclinical. What do we expect for the future? What are we aiming for? Let's go backwards a little bit. Backwards is here. Yeah. There are many, many attempts to treat neurodegeneration with many different drugs and with many different compounds. You can imagine the field is full of clinical trials that all of them have failed in different aspects because of toxicity, because of safety, because of a combination of both. There have been many, many trials that have been conducted in Alzheimer's disease, in Parkinson's disease, in multiple sclerosis, etc., with antioxidants, with antioxidative compounds. You know that's very much infection. We have to prevent oxidative stress in our neurons. When we block oxidative stress, we can have more healthy neurons, etc., etc. But unfortunately, all the trials that have been conducted with antioxidant compounds, for instance, with different vitamins, vitamin C, vitamin E, with coenzyme Q, with N-acetylcysteine, with different ROS scavengers, etc., have failed. They work very well in mice, but they don't work in humans. Why? We don't know. But what is true is that all the antioxidant trials that have been conducted so far didn't work. We have also tried many different clinical studies with compounds that prevent neuroinflammation. Neuroinflammation is also considered a bad prognosis factor in neurodegeneration. We There have been many, many different attempts with COX-2 inhibitors, for instance, with minocycline, with PPR gamma agonists, etc., and all of them worked well in mice, but they didn't work in humans. A little bit the same has occurred with anti-excitotoxic compounds, anti-glutamatergic compounds, memantine, riluzole, etc. They work very nicely in mice, but they don't work in humans. So what is the hope for cannabinoids to work in this devastating scenario? Little, but we have to try. And the optimism, I think the main reason for optimism is that so far we have used compounds that target neuroinflammation, compounds that target excitotoxicity, and compounds that target oxidative stress. But cannabinoids share the three properties. They have a broad spectrum action, so they are able to simultaneously target neuroinflammation, target excitotoxicity, and target oxidative stress. So maybe with these compounds, we have the three actions combined, and maybe in the future, we will be able to show that they target really neurodegenerative diseases in humans. Not to cure, of course, that is not possible nowadays, nor with cannabinoids, nor with anything, but at least to try to decrease the decline, the onset, the course of the disease. So this is the hope for the future, and I don't have the answer, but maybe in 5 or 10 years' time, I will be back here and I will show you positive results in that respect. So overall, we can say that cannabis are neuroprotective dots in mice, but we don't know whether they are or not in humans. We have to make the clinical studies, and as I told you, the hope is that they maybe can combine different positive actions to have most chances of success compared to previous therapies. And of course, it is very likely that cannabinoids are not so effective when used alone. Maybe they are effective when combined with other different interventions. So I think that is general for cannabinoid pharmacology. Usually cannabinoids in many different diseases, they have a moderate efficacy, but they are very good in synergizing with other therapies. So maybe if in the case of neurodegeneration, we combine this broad spectrum cannabinoids with other therapies that can target other different processes of neurodegeneration, we can have reasons for optimism. But so far, good news for mice, but question mark for humans. Thank you.
So we have a lot of questions.
Oh, yep.
So there's a selection of 4 questions. And may we ask Patricia Silveira to raise the hand? Oh, right over there, you can see.
Yep.
So it's in Portuguese, the question, and it says, are there any studies made on the plant in what concerns the treatment of the degenerative disease of the frontal lobe?
Yeah, well, as I told you, I said before, there are still no clinical trials that have been conducted with either isolated cannabinoids or cannabinoid combinations or plant extract for neuroprotection. The trials that have been carried out so far in neurodegeneration with cannabinoids have intended to palliate symptoms. So we have tried to decrease anxiety, to decrease spasticity, to decrease pain, to decrease nocturia, to decrease stress, to improve insomnia, etc., etc. But so far, there have not been studies in which, I mean, the trial has been sufficiently long and with endpoints that have been very well defined to assess neuroprotection, that we can really answer that question in particular, and the question of whether cannabinoids are neuroprotectants in general. There is only one trial that was conducted for that purpose. It was a trial that was conducted in Great Britain, in UK, some 8 years ago, something like that, 10 years ago, for MS, for multiple sclerosis. It was called the Cupid trial. It was a trial for 2 years, but with a very low dose of THC, and only THC, not a whole plant. And the trial showed that overall, THC at that very low dose, and it was only pure THC, as I tell you, was not effective to decrease the decline of, to decrease the progression of MS. But when the patients were substratified, it seemed that there were some subpopulations in which cannabinoids were kind of effective, whereas in other they were not. But when you did all the combination, I mean, the overall study was a failure, didn't succeed. But that trial, everyone knows that it was not correctly designed. It had several pitfalls. It was pure THC, not THC with other cannabinoids, or in a whole plant combination. The patient population was not very homogeneous and not very well defined. The THC dose was very low. It was from 2.5 to 5 milligrams a day. That is likely low, etc. So, I mean, we don't have information yet. We have to do clinical trials, and to do clinical trials with the proper cannabinoid combinations and in the proper patient populations to try to infer whether they can act in these neurodegenerative diseases.
Thank you. Shirin Matamed? Yeah, again, the most popular question asker in the room. You win the prize today. Is the method of ingestion administration important? And a secondary follow-up, when you referred to cannabinoids, are there particular cannabinoids that have shown to have more neuroprotective properties?
Well, that's a good point. I would say that depending on the disease and depending whether neuroinflammation, excitotoxicity, etc., is more or less important, then different cannabinoids have been shown more or less effective. Always in mice, because in humans, as I told you, there is no information about possible neuroprotective action of cannabinoids. In general, when there is more oxidation and oxidative stress, CBD seems to work well. When it's more excitotoxic, it's essentially THC, because it's a CB1-mediated mechanism. When it's microglial activation, it's usually THC rather than CBD. When I say THC, I mean CB1, CB2 receptor agonism. And when I say CBD, I essentially mean antioxidant action. So usually when one combines THC and CBD, or say CB1, CB2 agonists together with cannabinoids that do not have that property but that have antioxidant properties, antioxidant effects, usually the effects are better. So at least in theory, it would be better to put a combination of cannabinoids with different pharmacodynamic action than rather a single cannabinoid. Yeah, there have been many studies on THC, on CBD, on synthetic cannabinoids, also inhibitors of endocannabinoid degradation, so you increase the tone of endocannabinoids, and therefore you have more endocannabinoid signaling. There are a few studies, unfortunately, we know pretty well THC, we know so-so CBD, but the rest of the 130 cannabinoids, we know nothing, essentially. There is, I think, 1 or 2 studies on cannabigerol and some cannabigerol derivatives, and that's it. I mean, we don't know anything about CBDA, THCA, we don't know anything about THCV, CBDV, cannabinochromin, etc., 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 etc. So the world of cannabis is very TH-centric, and now it's becoming a little bit more CBD-centric. But besides THC and CBD, we have no information.
Okay.
Thank you.
Thank you. So we have another question, Sophia Tellers. Okay. So besides there are no studies that prove the efficiency against Alzheimer's disease, to whom has historical situations in the family of that disease, do you consider that the use of cannabinoids can be done as a prophylaxis of the disease?
Mm-hmm. Well, that is also a 1 million question in the field. I would say we don't know, but conceivably, why not? Yeah, but conducting clinical trials in that respect is very complicated, as you can imagine. A standard clinical trial with a selection of patients, etc., and their criteria for inclusion, exclusion, etc., is always very complicated. But when you talk about prevention, there are so many different habits, there are so many variables that it's extremely complicated to have an homogeneous population and a controlled population. So I wouldn't say no, but I cannot say yes. It's something to be studied, of course. There are good hopes for that, why not? But when talking about prevention, I mean, a pharmacological intervention, by definition, I would say it has to be dirty. Dirty in the sense that it's broad spectrum. And in that respect, cannabinoids fulfill that question. Because by definition, habits and all the preventive actions that we can do for preventing disease must target many different parts of health, because we cannot predict how our health is going to be in the future. So we want to prevent diseases. We, in theory, have to prevent all type of diseases. So we have to take care about our cardiovascular system, about our brain, about our liver, about our lungs, etc., etc. So we have to do, by definition, broad spectrum habits and broad spectrum interventions. So in that respect, yeah, cannabinoids are dirty drugs because there are many compounds with many different targets in the body. So they can be useful maybe for at least to have some hope on that. But yeah, it's an extremely important question. But I think first we should try to see whether in defined clinical populations, patient populations, we have some signals of action, and therefore we could go to prophylaxis. Of course, this is not something that I'm not saying at all what patients or individuals will do. Of course, everyone is free to try. But I think it's very difficult to say now from a scientific perspective. Yeah.
Thank you. Next question, Rafaela Pimenta. Thank you, Rafaela. Are there some studies in humans about cannabinoids and biological treatments that will work together as a neuromodulator or neuroprotectors with certain diseases like multiple sclerosis?
That's also a very good question, and the answer, unfortunately, is no. There have been many trials in MS in which cannabinoids have been given to the patients as palliators, of course, to decrease muscle spasticity, to decrease pain, etc. But usually those trials have been very short, usually from 6 to 12 weeks at the most. So it's very difficult to say in those trials. I mean, you can say something about symptoms. You can see whether the patient feels more or less pain, whether rigidity, muscle rigidity is improved or not, whether the patient goes to the toilet in the evening 3 times, 4 times, 1 time. That is simple to measure in 4, 5, 6 weeks. But talking about the efficacy of a treatment, and especially whether cannabinoids in combination with other treatment has been able to decline the course of a disease that can last 20 years in only 2 months or 3 months. I mean, the design of those experiments should be very different. And unfortunately, we don't have the trials, we don't have the information. Hopefully, Tilray or others will do in the future, will sponsor. This will be an enormous trial. Imagine, when you go for symptoms, you can go for 4 weeks or 5 weeks, 6 weeks, 2 months. But when you go for progression, you have to go to 2, 3, 4 years, and that is very complicated. But we have to do, because otherwise we will never know. And this is a very challenging field of biomedicine, and we have to aim for it, I think. Yeah.
Thank you very much.
Thank
you very
you.
much,
Thank you.
Doctor.
Thank you.
Thank you.